magic lamp, no matter what you ask for, the genie will provide you with it. It's a universal generator, the golden home of a genie who grants 3 wishes to the lucky owner of that treasure. What would you ask for? A hot lover? Money? Maybe winning a gold medal in the Giant competition? But what if you were cold? If you were thirsty? What if you were ill? The aim of our project is to develop the synthetic biology version of a magic lamp, a multi-purpose drug biofactory that is a system able to produce one medicine when and where you need it. We are the UPV agent team, and we are introducing you a DNA. But step by step, have you ever wondered how does a magic lamp work? The genie is the best wish maker ever known, and he has the information to make possible all your wishes. When you tell him your wish, he decodes the necessary information to make it. Along with that, he still needs the factory where your wish will be produced. And then, pop, happiness everywhere. So, in order to implement this in biology, we need a lot of imagination to find these equivalents. For example, first of all, which should be our wishes? We know we could ask for anything, but is there anything more needed and desired than health? Of course not. And that's the reason why, after doing research with charities, and especially Pharmamundi, we decided to produce 4 drugs which are very demanded in underdeveloped areas, in order to fight against cholera, viral diseases such as hepatitis and herpes, pneumonia, and diarrhea. Then, where should we keep our genie and let him produce our wishes? We wanted a robust and reliable factory, capable to offer plenty of resources. Moreover, it must also provide the physical integrity of our system. Ready for the answer? Seeds. Seeds are one of the most natural structures, tested by nature during millions of years. However, it doesn't matter how much you ask for things to a plant. You can cry, sing, or implore your wishes. It will not obey you. We had to translate spelled wishes to some language understood by those organisms. We had to implement inputs able to switch on and off the decoding of our medicine. An awesome idea illuminated our minds, literally, optogenetical control. Use of light has many advantages. First of all, it avoids the invasive use of chemical compounds. It can be controlled remotely, and it's friendly user. And last but not least, we needed our main component, the genie, the one who processes and keeps and stores information, the one who engages wish and reality. DNA. DNA is the ideal information keeper, and it can process it very efficiently. It must be our genie. So, the steps in order to create our SynBioGene lamp were, first of all, the design of our genetic circuit. Then, its testing in silico with our modeling task. After its approval, we carry on building it in wet lab. And finally, we create a real device and analyze the social repercussions of our project.
So now, let's start with the design. We have created a DNA construction that is controlled by the light. So we used photo switches in order to provide our genie with control. We started with 2 really understood devices that are ASLOF and phytochrome B. We also designed our brand new photo switch that is K-trompa and trompa. This works by when it is stimulated with the correct wavelength, the photo switch just joins and allows the transcription of the genes that are downstream. So after that, after implementing this in our seeds, we started with we had to design a genetic device. We created a kind of light code that is from ones that can be like a kind of binary code that acts as ones or zeros, and acts as one or zeros, so that with 2 stimuli you can produce 4 different outputs. As you can see here, the products we have selected are the 4 drugs. So, after struggling a lot, thinking about the device, we finally came up with the ideal solution. the ideal solution is our circuit, our Genie. We started producing constitutively the PFib and the ASLOF, so that allows the cascade reaction to start. After starting with this, this produces the recombinases with a light pulse. So this recombinase introduces memory in our system and also produces the PIF and the ASOF again, but with another binding domain. This allows to, with another light pulse, for example, this is a red pulse, production of the desired output. In fact, this output will be the drug we have selected. So this hasn't been proved in higher organisms, and this is not sorry. So we couldn't just go into the wet lab and start making stuff. We just needed to go from a very strong modeling in order to produce this. So here, my mate is going to show you how the modeling works.
Okay, so modeling, like your name, in tons of equations and coffee. And so we know that all those equations are proportional to the loss of your attention, and we'll go directly to the results. The aim of our modeling task was analyzing the critical points that could compromise the feasibility of our circuit. They were, first of all, which were the best light sequences, if the circuit will work under unusual conditions, and finally, if the new violet toggle we wanted to implement in plants would compromise efficiency. So we consider eligibility when the ratio between the desired output and other ones is higher than 100. And we perform simulations with different dark periods between both stimuli. Results concluded that continuity is preferred instead of two separate inputs. Thus, sequences blue-blue and red-red should be a single red and blue pulse. Continuity is also preferred with two colors in the sequence, and the length of the first irradiation is determined by recombinases, which are the key element. Then, will it work under unusual conditions? We perform simulations with triple sequences and simultaneous. Although results obtained didn't get one output being majorly produced, we get a mix of different outputs with ratios between them under 100, which could be useful for multi-production. And finally, the implementation of the new violet light toggle switching plants had the issue of the tetramerization of one of its components, which could lead to an inefficient activation. In simulations, T4 represents the percentage of tetramerization of this component, and results showed not only that this idea was possible, they also showed that with red-blue and blue-red sequences, the result was optimized. So, our model has tested the functionality of our circuit design, helped us to better understanding and optimize its performance, and finally, it discovered us new possibilities of production. Now we were ready to encourage our wet lab mates to carry on with its implementation in the laboratory. So go wet lab.
Thank you. Now, with the approval of the modeling team, what we have done is to implement our circuit inside the wet lab. The thing is that we are working with plants, and implement the whole circuit in a summer in a plant is something impossible. So what we did was a transient expression as a proof of concept of each key element in our circuit. Let me introduce you the working scheme with these elements. The first thing to do was build assembly, and it was made with golden braid assembly. Then pieces were transformed in E. coli to obtain a high copy number of each element. Then they were transformed in agrobacterium, a bacteria able to introduce our genes inside a plant. For that purpose, the agroinfiltration, where the solution with agrobacterium is introduced inside the leaf with a syringe. Then, in some occasion, we also needed to extract protoplasm, which are cell plants without cell membrane, in order to perform some of our experiments. So going back to our circuit, the first element to face were the light switches. We have proposed 3 light switches, one controlled by blue light, one controlled with red light, and one from our own design controlled with violet light. The construct with the blue light had 19 basic parts and more than 80,000 base pairs. It was constructed successfully, and we introduced it in our plant. Results show that after 48 hours, the expression was a little bit increased. However, this needs optimization and further studies are required. In the case of the red light switch, we perform the same construct with 19 basic parts, and in this case, it takes 19,000 whiskers. The results were performed in protoplast, and as you can see, after 13 hours, you can observe a differential expression between the cells kept in red light and those kept in dark light. These results were positive and were also the same for another binding domain that we described. And finally, we performed the same construct for our violet light. But this time, as it hadn't been proved before in plants, but we wanted to know if it was produced in the correct place of our cells. So we implemented in protoplast and looked at cells under the microscope. As you can see here, the nucleus is only visible when it is illuminated with the correct wavelength that activates the toggle switch. Only when the wavelength is not the correct one, the nucleus is not visible. And so we can show how we can turn on and off the switch. However, this needs further investigations, and we have to implement it with our production reagents. Now, the other key element of our circuit are recombinases. We have produced two different recombinases, BXB1 and FIC31, in our plants. And in order to prove their functionality, we have created a construct with the transcriptional unit of each of the recombinases and a reporter transcriptional unit. In this case, we have added a terminator between the promoter and the CDF region. So only if the recombinases are active, the GFP will be expressed. In these photos, you can see how both recombinases have worked, and they have a higher fluorescent levels than the backgrounds obtained from the negative controls. As you can see also, BXB1 was higher efficiency than FIC31. And now it's time for our wishes. Do you remember our wishes? We have construct 4 medicines construct, transcriptional units, and we have checked the production of 3 of them in our plant. As you can see here, with immunodetection, we have obtained we have detected the production of the 3 of our medicines. This is the antibody against rotavirus, this is the vaccine against cholera, and this is the antiviral. Now, we have all the information codified, but we needed something in order to decodify this information.
So now that we have the information inside the gene, we need to decode it in order to take our wishes out. At the beginning of this presentation, we have talked about magic lamp. You probably thought, what a crazy idea. Magic lamp doesn't exist, right? Well, we take profit of our multidisciplinary team, and we do not only design a biological circuit that acts as a magic lamp, but also brought the magic lamp with us. So basically, this magic lamp is a miniaturized bioreactor that allows us to germinate seeds inside of it. There are two different color lights inside under the lid that allow us to produce any different product that we want at every moment. Basically, this device makes our project portable and accessible to everyone.
This is an amazing project, and there is really not enough time in a 20-minute presentation to explain how much work has been put into it. So I'm going to start telling you a little secret. I am not a biologist. I'm a computer scientist, and I have no idea what's going on here. And do you know who doesn't have an idea of what's going on here either? Anyone else? High school students and children that want to get interested and begin to work with biological engineering have a hard time starting with all the lab procedures and handling the new environment. The lab is a very serious environment where your actions can have very serious repercussions. Because of this, we were thinking of finding an intermediate form of expression where you can actually, where we could get people introduced to the lab procedures. The thing is, to get people experience in the lab, you need a lot of time, a lot of money, and a lot of very expensive machinery. And all of this can go very, very wrong. So how do we get this medium in between? Using Minecraft technology, we were able to synthesize our lab, and not only our lab, this whole project has been replicated exactly. As you can see, now we're doing an assembly with the promoters and a CDS and a terminator to make the assembly of a transcriptional unit, just the way you would use in a normal thermocycler, only much, much faster. Now we're going to electroporate our final element, instantaneous action, as you can see. Just as in real life. And here we have our Petri dishes that doesn't react if you don't electroporate it, but if you put the correct element, you immediately get the blue and white colonies you will find in a normal Petri dish, only immediate. After that, you can pick the colonies and infiltrate our plants just the way you will do it in real life. And if you were listening to the presentation using different light patterns, we get different results only in the exchange with the coloring using colors. We can not only use this to simulate our project, but with your help we may be able to reproduce your project, making it more accessible to us and to everyone who wants to get it started. Not only that, as you can see, we've also made a replica of our building back in the Technical University of Valencia. This helps to get the user more familiarized with the environment beforehand, so he doesn't get as shocked at the beginning.
For the Applied Design Special Prize, we have designed and implemented a portable bioreactor. Moreover, the whole project is inside the integrated human practices, since the whole project is based on making medicines portable and accessible to everyone, everywhere. Finally, for the educational and public engagement, we have created a virtual lab called SynBiograph, in which everyone can learn synthetic biology while playing. We have also collaborated with a lot of teams, and we have submitted 14 parts to the registry. And finally, we want to thank to all the people that made this project real and possible, and mention all the team members that worked really hard this summer. And just to end, I want to remind you the main idea of this project. No matter where you are, no matter what you need, just ask your wish, because our DNA has no frontiers. Thank you.
Thank you very much for a very engaging presentation. Are there questions from the judges?
So, okay. I love SynBio Minecraft. It cracks me up. Did you get anybody to try it outside your team?
Yes.
What happened?
It worked well. I mean, if they know how to play Minecraft, they are able to play SynBiograph. It's very easy. Just follow the steps on the guide we have created, and it works well.
Who's the they? Trying it?
We have 2 high schools that have agreed to use SynBiograph to teach biological engineering to. We have 2 high schools that have agreed to teach synthetic biology using our method. Already in Valencia, we have a didactic guide already written. We have 3 practices, and we are going to actually go to these schools, I think, next month.
All right, other questions?
Yeah, I just first of all, that's great. I just want to follow up that question. Does after they've played, what is it called, SynBiograph, can you ask them questions about the actual synthetic biology? So in other words, can you get them to translate the SynBiograph knowledge sort of back into a superficial level of understanding? Okay, go ahead and try it.
To start, this model that we've built now is only doing the practices and having an input-output research. On the near future, we can implement anything from a questionnaire to what would you do with this element to anything that you can think
Coming back toward the LNDNA, something I was a little bit unclear on. So I understand the idea that you expose the materials to the light and start the reaction, but then the actual drug production, it sounded like the idea is that there's a seed in there which you germinate, and then the plant grows inside the bioreactor, and then somehow it purifies this to get a drug of high enough quality that a person would be willing to take it?
It depends on the drug that you are going to take. For example, the cholera vaccine is an edible vaccine, so you can eat it. If the plant is a plant that you can eat, it might be a way in order to take it. But on the other cases, you can tag your protein in order to produce it in the apoplast part of the plant, and with a pump, with a vacuum pump, you can take the medicine from inside your plant, and in that way you just have to centrifuge it, and you have the purified medicine.
That fits in the lamp too?
This is the first prototype.
Question?
Just following up, and apologies if I missed this, but were you able to grow anything in the bioreactor? Did you try?
Well, we tried to grow soya beans also and spinaches, so they worked, and I think that everything was filled okay.
Cool.
So if you grew something, how long does it take from you actually pressing the button to actually getting a drug that you can actually use to treat the disease?
It takes around 50 hours to complete all the process, from the first light to the last light, more or less, as the modeling calculated.
But with the modeling task, we have results are qualitative because we have been able to implement the whole circuit in the plant, so we don't have wet lab feedback, but with estimations from literature, we think that in more or less 2 days we can reach the production with ratios higher than 100 between the desired output and output.
So you can see the direction my question is going. Will the person be over the disease before they get the drug?
Yes, yes.
We recommend you to planificate your
You know, yes.
Any other questions from judges?
I guess the distributed production of drugs is sort of a hot topic within SynBio these days. I was curious if you discussed, the idea with sort of just the general public about like how do they feel about these capabilities getting out and being used by just normal folks.
When you explain to the people that if they just eat a plant, they can be cured, they get mesmerized and they love the idea. However, as you might know, in Europe the GMOs are not quite well known. And if you explain them that this is a GMO, then they change their opinion. But I think that it is because a lack of information from them. That's why that's one of the reasons why we created SynBioCraft.
Talking of GMOs and EU regulations, did you have to individually or as a lab get certifications to be able to do this work?
The lab.
Can you repeat the last part of the question?
Because of the EU directive about dealing with GMOs, did you each individually have to be certified to handle GMO materials, and did the lab have to be certified to be able to do this work?
Our lab, of course, has the corresponding permissions to work with this kind of plants. However, we made transient expression, so we, in fact, never made a transgenic plant, so that's not a problem. In the case that we want to make our seeds, put them to the market, we will have to make the studies and follow the protocols of the European Union to make the seeds approved.
All right, well, let's thank the team one more time.